Hi everyone. In this video we're going to talk about cardiac muscle contraction, and in order to do so we need to review and expand upon our previous discussion of cardiac muscle tissue. So this drawing should look a little bit familiar. What I'd like to do is take a cross-section of the cell and draw in some of the structures so that we can start to see them in 3 dimensions. So first we have our sarcolemma, which is the cell membrane of a muscle cell. And inside of the cell is many myofibrils. And myofibrils are bundles of filaments, particularly actin and myosin. And surrounding the myofibrils are the sarcoplasmic reticulum, which is comparable to endoplasmic reticulum in other cells, but in muscle cells we call it sarcoplasmic reticulum, and it's really notable for storage of calcium. We also have these structures called T-tubules, and the T-tubules are basically like tunnels that are sort of throughout the muscle cell. And then we also have abundant mitochondria. So let's just draw the myofibril, and we'll sort of organize the filaments that are contained within the myofibril so we can become a little bit more prepared to talk about what happens during muscle contraction. So in orange we have myosin fibers, and the myosin, which we mentioned before is a protein, has these little sort of heads that come off of it on either end of the myosin. And eventually we're going to use the myosin head to interact with the actin. So we'll use that structure to allow for muscle contraction. The green is actin, and actin is basically there are multiple forms of actin, and I like to think of actin as kind of like a bead of pearls. And in this drawing I only drew one sort of chain, but actually actin is sort of 2 intertwined chains of pearls. And then it's surrounded by, in blue, tropomyosin, and attached to tropomyosin is troponin. In red we see titin. Titin is another type of protein that actually sort of anchors the myosin to itself, and then also connects to actin along what we call the Z line. So the myofibril is actually very long, and we sort of break it into segments, and we actually divide the segments by Z lines. So between one Z line and another Z line, we call that a sarcomere. And so that's basically like the functional unit of the myofibril. And so we can sort of think of the muscle shortening based upon this particular organization from Z line to Z line. Now the M line is like the midline of the myosin, and then we have several bands. And so the H band is basically the space between the actin filaments, so it's just made of myosin. And then there's an I band, and the I band is the space between the myosin filaments where there's just actin. And then the H band is kind of in its own little category because we actually sort of see an alternating pattern. In between all the I bands is A band. An A band contains the H band, but basically it's from the point of overlap of the actin and myosin where that begins all the way till it ends for the myosin filament. So basically it's like the entire length of the myosin fibers. And so we see this alternating pattern: I band, A band, I band, A band, and so on. And so what I just want to point out is that if we'll just draw this a little bit smaller up here, we can actually see that as the I band and the A band sort of alternate, we get this sort of pattern, and it actually looks like stripes. So you notice that in this picture, the filaments, actin, myosin, are in the horizontal direction, but the stripes are actually in the vertical direction. And so the stripes is what actually we refer to as striations, and that's where we get our striated appearance of cardiac muscle tissue. Okay, so now let's draw the myofibril again. In this drawing, I want to talk a little bit more about the T-tubules that we saw in the previous drawing. And so in cardiac muscle, we see we are still organized by sarcomeres, and then in between each of the sarcomeres is sarcoplasmic reticulum, and that basically surrounds the myofibril. It's cylindrical in its conformation, and then we'll just draw in the cell membrane, and we'll see that the T-tubules sort of extend along the Z line. And so it's a little bit difficult to depict in this particular drawing, but I want to sort of convey that the T-tubule will sort of encase that particular section of the myofibril, but also extends to the cell membrane. So technically everything that's sort of within the T-tubule tunnel is extracellular fluid. Okay, so now let's just sort of pretend that we're taking a slice along the Z-line, and we'll just draw a cross section so that we can look sort of at it head on. So we can see multiple myofibrils in this particular cell, and the myofibrils are surrounded by the T-tubules. What we get basically is this sort of network of tunnels. So I just want to reiterate that because this is a cross section at the T-tubule, if we were to take a cross section at a slightly different place, we wouldn't see exactly this picture. We would see something slightly different. So this is just sort of one view at the point in space where we decide to take this to make this drawing. Now let's take this same original drawing and let's actually slice it right down the middle. It's almost as if we cut right into the middle of the myofibril. So you can see the T-tubule still extends above, and because we cut a cross section here, we're not able to see that the T-tubule would actually sort of come out of the page and then still surround the Z-line of the myofibril. But since it's no longer three-dimensional, we're just drawing it flat. We'll just draw the Z-line right in the middle. And then we have, of course, our myosin filaments and our actin filaments. And now let's also just take sort of a cross-section of our sarcoplasmic reticulum. So it's kind of like a web-like structure, and so just sort of envision everything that's kind of within the purple lines is going to be sarcoplasmic reticulum, and then within the spaces in the middle will be ECF. And so this is a little bit of a cartoon drawing, but I'm just hoping that we'll kind of get the point across. So what I really want to convey more than anything in this picture is the proximity of all of the structures, the T-tubule to the sarcoplasmic reticulum, and then also to the myofibril. And that's going to become important as we explore the relationship between each of these structures. And then lastly, let's just zoom in on the T-tubule and the sarcoplasmic reticulum. And one thing I want to mention is that we know already that calcium is in high concentration in the ECF. So there is a high concentration of calcium within the T-tubule. And within the sarcoplasmic reticulum, there's also a high abundance of calcium. Major function of the sarcoplasmic reticulum is storage of that calcium. And that's actually because we need the calcium for muscle contraction. So let's go ahead and draw a couple channels in the membrane. So there is a voltage-gated calcium channel in the membrane of the T-tubule. And this voltage-gated calcium channel is sort of associated with several receptors, including the dihydropyridine receptor, or DHPR. And one thing that we really haven't talked much about yet is the potential for a voltage-gated channel to be blocked in some way. So we've mentioned, that there could be an agonist or an antagonist that could block a ligand-gated channel, but we really haven't mentioned how this could happen in a voltage-gated channel. So what it will be easiest to envision is this channel is more or less cylindrical or three-dimensional in structure, and so there's a sort of a tunnel in the middle, and it's not different just because it's a voltage-gated channel. And so somewhere within the inside of that channel is actually a receptor, and that receptor, if something binds to it, will there's a couple of sort of ways to look at it. One is that there's now physical obstruction that's preventing the ion to pass through, and then also the sort of thing to consider is that how it binds, it could stabilize the particular channel in an inactivated state, and so therefore it just isn't really able to open in response to depolarization or hyperpolarization, whatever the particular channel is sensitive to. And so in this particular case, I really mention this receptor because this is the receptor that's blocked by certain types of calcium channel blockers, including diltiazem. Now we also notice a second voltage-gated calcium channel that's present in the membrane of the sarcoplasmic reticulum. And this particular voltage-gated calcium channel is associated with the ryanodine receptor. And the ryanodine receptor is of particular interest to us because in skeletal muscle, so it's not true so much for cardiac muscle, but in skeletal muscle, certain types of patients might have a mutation in the ryanodine receptor where they have a significant increase in the calcium that's able to flow out on account of stimulation, and this is what leads to malignant hyperthermia. So that's a disease process that is discussed, in conjunction with certain medications succinylcholine. All right, so now we also want to draw in a couple of transporters in the different membranes. So let's draw in a transporter for calcium in the membrane towards the sarcoplasmic reticulum. So this particular transporter is ATP dependent, and so we'll put ATP in the middle. It transports calcium. It is a pump, so it's moving calcium against its concentration gradient from the cytosol into the sarcoplasmic reticulum. And this transporter is actually called SERCA. It stands for sarcoendoplasmic reticulum calcium ATPase. But it's really just, think of it as just a calcium pump that pumps the calcium back in. And so this is important because when we pump the calcium back in, we make the calcium available for the next contraction. So if we want to affect the rate of contraction in the heart, what we would do is we would utilize medications that will cause a number of effects, including faster reuptake of calcium so that we can get faster depolarization and contraction the next time around. We also have just a plain old calcium ATP pump that pumps the calcium out of the cell into the ECF. We have a sodium-potassium pump, which we're already familiar with. And last we have NCX, a sodium-calcium exchanger. So this particular transporter does not utilize ATP. Instead, it actually functions by secondary active transport. So the sodium gradient is high concentration of sodium outside of the cell, low concentration of sodium inside the cell, so the sodium is able to move down its gradient. And we're actually moving in 3 sodium for every 1 calcium that's exchanged in this particular transporter. So as the 3 sodium ions move in, 1 calcium ion is moving out, and so it's harnessing the energy from the movement of sodium in from down that gradient in order to transport the calcium out. Okay, so now let's talk about what happens during depolarization. So we know that cardiac muscle cells are connected by intercalated discs which contain gap junctions, and so the way that the cardiac muscle cell is going to be depolarized is by way of sodium movement through the gap junctions from one cell to the next. So we can see the sodium that moves in along the membrane, and as the sodium reaches the first voltage-gated calcium channel in the T-tubule, we can see that the voltage-gated channel will be activated and calcium will be able to move in to the cytosol down its gradient. And then the calcium now is increasing the positive charge in this vicinity, which is also near the second voltage-gated calcium channel that is present in the sarcoplasmic reticulum membrane. And so we have a process called calcium-induced calcium release, meaning as the calcium is moved through the red voltage-gated calcium channel in the T-tubule, it activates the voltage-gated calcium channel in the sarcoplasmic reticulum, which involves more calcium movement into the cytosol. So it's sort of like a cascade of reactions. Calcium release causes more calcium release, but from a different source. So it's called calcium-induced calcium release. And so what happens after the calcium is in the cytosol is it floods its way down to the myofibril, and it's actually going to associate with troponin, which is present on actin. And so the calcium is basically one of the 2 major components that allow for muscle contraction to occur. We're going to explore that a little bit more in a moment. But what I want to mention is that we said we have all of these transporters, SERCA, the calcium pump, the sodium-potassium pump, and NCX all present, and their job is to remove the calcium from the cytosol and transport it either back into the sarcoplasmic reticulum or into the ECF. And so once this calcium floods down into the myofibril, it will associate with troponin for a little bit, and then eventually all of these structures are going to pump the calcium back where it originally started, and that will actually lead to muscle relaxation. So arrival of calcium is associated with contraction, and removal of calcium is associated with relaxation. And so we think about the non-pacemaker action potential. We recall that there is a plateau phase. It's all about calcium movement into the cell, and the sources are from the T-tubule and from the sarcoplasmic reticulum, so that we can have this association of calcium with the myofibrils. This is a really good demonstration of what's called excitation-contraction coupling. It's also you could think about electrical function versus mechanical function. Electrical function is all about the depolarization and making the calcium available, but mechanical function is about calcium's job in muscle contraction. And so without that first part, we can't have muscle contraction, but without muscle contraction, we don't really have any real cardiac function. So the two are invariably linked. Okay, so now to finish up our discussion on muscle contraction, let's just sort of draw the structures a little bit larger and take a better look at what happens in terms of the actin and myosin interaction. So we notice that we have actin, we have tropomyosin, and we have troponin, and those three are all connected. And then we have myosin, and we mentioned that the myosin has these heads that sort of extend off of it. And we're drawing this muscle in a relaxed state. So actin and myosin are not connected to each other, but the myosin heads are holding on to an ADP and an inorganic phosphate. So think of an ATP that's already been split, but the myosin is harnessing the energy from the split, from breaking off that inorganic phosphate, and it's holding on to both these molecules. And so there's one ADP and inorganic phosphate for each myosin head. And so let's just remember real quick that we have calcium that's coming from the sarcoplasmic reticulum. The calcium is going to bind to troponin. And so when calcium binds to troponin, there's a sort of a cascade of events that occur. So when calcium binds to troponin, tropomyosin moves off of the myosin binding site. So normally at rest, it covers the myosin binding site on the actin, and it doesn't allow for myosin and actin to interact. But when there's calcium available, the tropomyosin moves out of the way, and now there's a binding site on actin that's available for myosin to sort of grab onto. And as soon as that happens, the myosin binds to the actin. And so we'll just show it again, and this time we'll show the myosin and actin connected. And so when the actin and myosin are connected, we actually call that cross bridge. So cross-bridge formation is the act of binding actin to myosin, and so once they're connected, they're sort of we just say that the connection is a cross-bridge. So initially, as soon as this connection is made, the ADP and the inorganic phosphate are still attached. But next, what's going to happen is the myosin heads are going to sort of pull towards the center of the myosin. So remember, the heads are pointing sort of away from the center. So they're going to pull towards the center in what's called a power stroke. And so as they pull towards the center, they're utilizing the energy that's from the ADP and the inorganic phosphate split. And so as this energy is utilized, the ADP and the inorganic phosphate are released. I just want to draw a quick analogy to the idea of cross-bridge and power stroke, just to sort of make sure that this sinks in. So let's imagine like a canoe or like a crew, and then there's a bunch of people that are all sitting in the boat together, and they're holding all their paddles. And you can imagine that if they, especially, they're going to be sort of all synchronized, but they will all pick their paddles up at the same time. That would be the actin and myosin are not connected. They dig the paddles into the water at the same time. That's our cross bridge. And then they pull back in order to propel the boat forward, and that is our power stroke. And the energy is truly expended during that pulling of the water back to move the boat forward, and so that's when the energy would be released. And then what they'll do next is they will pull their they will pull the paddles back out of the water, they will reach forward, and then they will dig the paddle in again in order to move the boat forward once more. So that relationship is exactly the same as what happens in actin and myosin, except that in the case of the paddles coming out of the water, in order for the myosin to separate from the actin, a new ATP must arrive. And the new ATP is actually going to allow the release of myosin from actin, and then immediately after that, as the head is moved back into place, the ATP will be split into ADP and inorganic phosphate for utilization in the next time that the myosin head connects to the actin. So as long as the calcium is still available, the myosin heads will release, and then they will reach up and they will attach again. So this is like a sort of a continuous process. This happens like a couple of times in a row. And so we essentially are going to get shortening of the muscle. So remember that the myosin, when we looked at the original drawing, there was sort of like a midline for myosin, and on either side are these heads, and in both cases the heads pull towards them. And so it actually causes the shortening of the muscle fiber altogether. And so this process just sort of repeats over and over and over again until that calcium is taken back up into the sarcoplasmic reticulum and expelled from the cell into the ECF, and then we need a new depolarization in order for the next muscle contraction to occur. All right, so that concludes our explanation of cardiac muscle contraction, and I really recommend if the first couple of videos haven't come together yet, now that we've come full circle and seen muscle contraction, it's really very important to go back and make sure that there's a clear understanding of the relationship between electrical function and mechanical function, or conduction and contraction. That's all for this video. Thanks for watching.